Let me introduce the first speaker, who is Dr. Deborah Charych, I think. So, Charych. So, sorry. And Deborah is a founder and the CTO of RayzeBio. So, prior to RayzeBio, she had several scientific leadership positions in different biotech companies, including Nectar, 5 Prime, and Aron. So she used to work at Lawrence Berkeley National Lab as a principal investigator, which is very good. She received her PhD in chemistry from University of California at Berkeley. So she's going to tell us about the tumor-targeted radiotherapy by using peptides. Robert?
Thank you so much for that introduction, and appreciate the invitation to speak here at TIDES at this radiopharmaceutical track. Please note my disclosures. I am a shareholder and advisor for RayzeBio, a startup company in San Diego focused on radiopharmaceuticals. And I'm also on an advisory board for a number of other biotech companies. Despite the stellar advances in cancer treatment using immune therapies, it remains a stubborn disease, unfortunately, and it's still a leading cause of death and suffering for families and friends. We often think of radiation therapy as an old modality. In fact, there have been remarkable advances. External beam radiation is routinely and successfully used in a majority of tumor types, and the ability to target a lesion with an external beam or ray of radiation has improved significantly. However, external beam radiation doesn't work very well if the patient has widely metastatic lesions. In that challenging situation, systematically targeting and delivering radiation to the tumor has proved to be a valuable modality. And while several targeted systemic radiation drugs have been approved, and I've listed a few of them here, it felt like we were just barely scratching the surface. Conceptually, the idea of targeting radiation is somewhat similar to antibody-drug conjugates, except instead of a auristatin or DM1 payload, we are carrying and delivering a decaying isotope. The conjugate is injected by intravenous infusion. It literally homes to the tumor via the tumor-specific antigens on the tumor cell surface. And the radioactivity from the decaying isotope damages the DNA of the tumor cell, leading to tumor cell death. It sounds simple. What could go wrong? So to build a pipeline around this concept, it may be useful to think about the chemistry in modular units that can be replicated for different tumor targets or different tumor antigens. We may prefer reasonably well-validated tumor targets to minimize clinical risk. So at its most basic, we need a targeting moiety and some kind of connector, often called a linker, and a chelator cage, often termed the DOTA cage. And I've shown each of those components on the slide. And that DOTA cage will house the radioactive metal isotope. A key point about the isotope is that we can swap it. So that's the thing inside the gold ring there is the isotope. We can swap that isotope so we can have it be, say, gallium-68 for imaging a patient, and then we may decide to treat that patient with a therapeutic isotope, such as lutetium-177. It's just a different metal. And lutetium-177 is a beta emitter. The key concept is that we can use the very same conjugate that we've designed, the very same manufacturable unit, if you will, and all we are doing is chelating a different radioactive metal ion. The chelation of radioactivity is a separate manufacturing step, and at that point, your product will start decaying as soon as you make it, which may be a unique experience for those of us who haven't been in this field forever, especially from a process development and CMC perspective. Okay, so let's please start with that decaying isotope chelated into the DOTA cage. As an isotope undergoes its decay process down to the ground state, that atom is emitting radioactivity. The radioactivity is fundamental and in the form of subatomic particles, simplified as alpha, beta, and gamma on the slide. The important point is that alpha is like a cannonball. If you're a piece of tumor DNA and you're in the path of an alpha, it's not good for that DNA, which is good if it's a tumor DNA. The alpha particle is 4 orders of magnitude more massive than the beta particle. And the good news is that the alpha particle doesn't get very far. Its path is measured in microns. Actinium-225 is an example of an alpha particle that has been tested in humans. Now, beta particles is a flightier particle. It has a mass equivalent to that of an electron. Its path is measured in millimeters. It also works well in the clinical setting, and lutetium-177 has been extensively used in humans. Now, the gamma particle, the one shown at the bottom, and also positrons, I should add, they travel very far. So their path length is measured in meters. And that's so we can image inside a human that's in a scanner. Or we can also image a small animal. The gamma and positron particles are critical for enabling PET and SPECT imaging, which can significantly impact clinical development of new radiopharmaceutical agents. As an example, please focus on the 2 images on the left which are 2 images of the same man with metastatic castrate-resistant prostate cancer. In the first image, the PET image, gallium-68 was chelated to a conjugate that binds prostate-specific membrane antigen, which I'll call PSMA for short. PSMA is a tumor antigen that is highly expressed on the surface of prostate cancer. So I'll call that image on the left a PSMA PET image because it is antigen specific. The second image of this pair is a standard FDG PET, imaging glucose metabolism. Now please focus on the red circle. Hopefully you can see that red circle on the image on the left. The tumor mass is inside the 2 red circles. Now what is striking here is that by FDG PET, we can see a large bulky tumor mass. That's the image on the right. However, by PSMA PET over on the left red circle, we don't see very much at all, meaning that this patient's tumor does not seem to be expressing very much PSMA antigen. These important images allowed Dr. Michael Hoffman, the investigator in Australia running this clinical trial, to make the decision that this patient on the left, unfortunately, would not be a good candidate for the study. So we can enrich our clinical population with only those patients whose tumors express the target of interest without taking a biopsy. And Dr. Hoffman did just that and treated these patients with the very same construct used for the imaging, only this time, instead of gallium-68, the lutetium-177 beta particle was chelated. Now, in prostate cancer, there's a biochemical marker called PSA, not PSM. PSMA is the tumor antigen, PSA is the marker. PSA is a soluble protein and often highly elevated in advanced disease. If it drops by 50% or more after treatment, it's potentially a good outcome. I'll call this type of response a PSA 50 for short. In this study, 57% of men had a PSA 50 response, represented by the blue bars in the center waterfall plot. Now, as a cancer scientist, I've been a cancer scientist for a very long time, the ability to image the tumors non-invasively without asking the patient for a biopsy was hugely attractive to me personally, and the reason for wanting to work in this field and starting a company around it. So imaging is not only valuable for patient selection, as hopefully I demonstrated, but also valuable for translational development. We can study in detail the pharmacokinetics in the blood, as well as in the tumor, the pharmacokinetics in the kidney, the pharmacokinetics in the liver, the pharmacokinetics in the spleen. It's like PK on steroids. We can gain mechanistic understanding of the distribution and fate of our molecules and directly see the effects if we introduce a chemical modification. The example shown here is of a peptide targeted to an antigen called fibroblast activation protein, or FAP, expressed on stromal and some mesenchymal tumors. This is a pretty good in vivo drug profile in that the tracer is taken up quickly by the tumor and it leaves the blood pool fairly fast. It doesn't seem to be retained by any normal organs other than the tumor, and that's exactly the kind of profile one would like. So returning now to our modular conjugate, let's please focus on the targeting moiety, this piece over here, for which there are many options, and I listed several examples over on the right side of the slide. Oops. We are mindful that the field is not new, and many excellent approaches have been developed, for example, with antibodies and small molecules and cyclic or constrained peptides. The philosophy we took is to not necessarily be beholden to targeting moieties that already exist. Rather, we approach the problem from the clinical oncology and tumor biology perspective to select what we believed to be important tumor antigens. We then worked backwards and asked how best to target these tumor antigens. In other words, what should the chemical matter look like? While our desirable tumor antigens in mind, we knew that there were several. We did a bunch of diligence around bunches of different platforms. We decided to focus on the low molecular weight part of the spectrum as shown in this table. The last line of the table is a key line. That's the brown highlighted tumor target type. Because we wanted to be able to target virtually any tumor antigen. Similar to antibodies or antibody fragments, macrocyclic peptide binders can be found to just about any antigen. But like a traditional small molecule, we can make them synthetically in the lab and conjugate them site-specifically. So that was a key advantage over the biologics. The other reason for focusing on the low molecular weight regime is that smaller things tend to be more tumor penetrant. The images on the left are of a three-dimensional tumor cluster called an organoid, or tumor cluster. The constructs are labeled with a green dye, and each column going from left to right represents increasing molecular weight from 1 kilodalton to 300 kilodaltons. The lower molecular weights appear to fully penetrate the tumor. However, if you look above about 10 kilodaltons, the construct starts to get stuck on the outer edge of the tumor mass. So you can see that's sort of this ring of green that's lighting up at around 50 kilodaltons and 300 kilodaltons. So this type of data actually agrees pretty well with the mechanistic modeling data shown on the right from the Wittrup lab, not far from here. The U shape arises because monoclonal antibodies, as you get higher in that U curve, that have FcRn recycling. So if you wait long enough, there will be tumor uptake just from the extended exposure. Now, whether at the high end of the antibody spectrum, whether they fully penetrate or form a ring, similar to what we see in the images, I think is not completely clear. I would argue maybe they won't penetrate as deeply. So a design goal is we'd like to go about less than about 10 kilodaltons. That seemed reasonable. The other potentially important aspect of low molecular weight constructs is their rapid pharmacokinetic profile. On the left is a typical PK curve for a monoclonal antibody. On the right is one of a peptide, and this is just a random peptide from the literature. The peptide clears the blood pool very quickly, which may be an advantage if you've got a decaying isotope on your tail. Of course, we'd want to also quickly get taken up by the tumor and stay in the tumor and give maximal exposure, or AUC, in the tumor of this radiation. So another potential design goal is rapid clearance from the blood pool to minimize exposure of the bone marrow to the radiation. Now, it's interesting to look at this with two concrete examples, both targeting the PSMA tumor antigen on prostate cancer cells. And On the left is a monoclonal antibody to PSMA known as J591. The targeting moiety is essentially the CDR domain of the antibody. On the right is a small dipeptidomimetic called PSMA 617 that binds the PSMA catalytic domain. It's a membrane enzyme. And this PSMA 617, the binding region, is actually a mimic of the endogenous PSMA enzyme substrate. The difference is about 150 kilodaltons for the antibody and about 1.5 kilodaltons for the PSMA 617, or the dipeptide. So this is about a 100-fold difference in molecular weight. The PK half-life of the antibody is about 39 hours in humans, compared to less than 30 minutes for PSMA 617. Now, imaging studies in humans shows a fuzzier image of the tumor lesions for the J591 antibody, and this image, that's the image on the left, was taken 7 days after the patient was injected with the tracer. So the patient has to return to the clinic 3 to 7 days later just to get an image to see if they are eligible for the therapeutic isotope. On the other hand, shown on the image on the right for PSMA 617, this is an agile small molecule. It's crisply taken up by the tumors in less than an hour, and the patient can be imaged the same day, and a decision can be made should they get the therapeutic isotope or not, just like in the Hoffman example. Now, the effects on clinical response and hematological toxicities between these two agents seems fairly striking. Using, again, the measure of PSA 50, or the 50% decline in the PSA as a response criteria, using PSA 50 as a biochemical response, please note that the waterfall plot on the left, where 11% of men had a PSA 50 response, the left one is for the J591 antibody, the one with the high exposure. On the other hand, with the waterfall plot on the right, 66% of men had a PSA 50 after treatment with the emitter, with the small PSMA 617 agent. Now, please note that both agents are chelated to the beta emitter lutetium-177. In addition, please look at the table in yellow below, which notes the percent of patients with a grade 3 or grade 4 hematological toxicity. For the J591 antibody, this rate is 46.8%. That's fairly high, compared to 15% for PSMA 617. Now, we know it is difficult to do these kinds of cross-trial comparisons, but these are fairly large effects, and they seemed notable. It is possible that the hematological toxicities simply limit the ability to dose the antibody to its maximum potential. So how can we build a platform around these ideas and a pipeline of low molecular weight agents to new tumor targets that go beyond PSMA and SSTR2? And I should say that PSMA and SSTR2 have been kind of mainstays of this radiopharmaceutical field. So in order to build a pipeline, we'd have to screen for new binding moieties to those new targets that we like, and do the harder work of finding chemical matter that has high affinity, ideally less than 1 nanomolar Kd, and is also highly specific to our target. We'd like fast kinetics for the reasons discussed, and the way to really assess how well your novel agent is behaving is to do radioactive biodistribution studies, usually in a mouse. So a key design feature of a binding moiety platform is that it gets you fast to the in vivo part. So we don't want to spend too many years in this circle of in vitro land. The platform should productively provide novel chemical matter that is high affinity and specificity so we can move over to the right and do the in vivo biodistribution. Now, there are many options for low molecular weight platforms. On the left is a typical small molecule, high throughput screening or medchem type library approach. In this case, the choice of target could be important because it may require that its target have a deep pocket for a small molecule. On the right are phage or mRNA display libraries of peptides. These are generally restricted to the 20 canonical amino acids, but the targets or epitopes may not need to be limited to those with a deep pocket. So a shallow groove may be okay, similar to the epitopes that an antibody might find, but the kinetics would be like a small molecule. In the middle, in orange, are approaches that combine small molecule diversity with the sheer numbers and permutations available using canonical, non-canonical peptide display on phage or mRNA. Now, constrained peptidic libraries are very interesting. And I've just listed two examples here. On the left is an mRNA display peptide library from Peptidream in Tokyo, Japan. That machinery, the biological display machinery, accepts non-canonical amino acids via a mutated ribozyme, which is pretty cool. Library sizes are on the order of 10 to the 14th, so the chances of finding something good are reasonably high. On the right is a light-directed synthesis on an array surface, which by virtue of being synthetic can accept non-canonical amino acids with library sizes on the order of 10 to the 7th. Now, this is probably just my opinion, but I would argue that chemical diversity of a very large library is an important parameter. The scatter plot shows chemical diversity on the y-axis and molecular weight on the x-axis for a non-canonical Peptidream library in purple and a bicyclic peptide library or canonical peptide library in green. Now, if you combine the purple, that chemical diversity that you can get with 10 to the 14 options, it would seem that, that low molecular weight, high affinity, high specificity to the target that you like can be found to just about any target. Importantly, it can also be found relatively quickly so that we can get out of that in vitro circle and quickly get into the all-important in vivo biodistribution studies. Ideally, we want to succeed or fail fast and then adjust accordingly. Now, in our own hands, we found that incorporation of non-canonical amino acids does offer, in fact, high affinity and specificity to multiple epitopes that we are interested in. And this, in fact, we've been able to go in vivo after just about 3 or 4 months, which is pretty remarkable. And I've been in this field for a long time, and I know how long it can take to optimize affinity and specificity. So we can, sometimes it can take more than a year to get to an in vivo study, but we were able to do it in just a few months. Turning briefly to the linker aspect of our modular design. The linker can be a simple connector, or it can serve as have a specific function. I've listed a few examples of linkers on the right here of the slide. The radiopharmaceutical literature has many examples on the use of albumin-binding linkers to extend the half-life and exposure of the conjugate. On the surface, this may seem like a good idea, but translation to the clinic can be challenging because increased exposure in the blood pool may lead to dose-limiting hematological toxicities, kind of like what we saw with the antibody. An example here is PSMA 617, whose structure is shown in the middle and on the top, and the same molecule, essentially, except with an installed albumin-binding moiety circled in blue in the bottom structure. The dipeptide is in green and the DOTA cage is in yellow. While the preclinical data at left are promising in mice, the issue is that in the clinic, which is some of the data shown on the right, the PSA 50 was 50% after 3 cycles, which is respectable and comparable to unmodified PSMA 617. But unfortunately, in this best response group in the clinical study, every patient had a grade 3 or 4 hematological toxicity. And half the patients on this study, 14 out of 28, had to discontinue. So this heme issue is an important point, I think, for the translation between mouse and human. So regarding the isotope, beta or alpha, there's no question that beta or lutetium-177 has proven itself worthy with the approval of Lutathera and excellent phase 3 pivotal data from this past ASCO. And PSMA 617, that was just this past ASCO in June of 2021. Now we also have the approval of Lutathera, but the alpha isotopes are compelling. It was compelling to me, at least, because they were about 1,000 times more potent than beta. They seem to cause irreparable DNA damage, meaning that that cell's going to die. The alpha seems to also work under tumor hypoxic conditions, which is quite interesting because beta seems to require reactive oxygen species to damage the DNA. But if the tumor is hypoxic, by definition, there's no reactive oxygen, yet alpha seems to work under those conditions. Now, we do have a lot to learn still about the biology and the tumor mechanisms involved with both of those processes, whether it's reactive oxygen or hypoxic. There's a lot of biology there. Now, in the clinic, the alpha agents are fairly interesting. It's fascinating that you can see a response with the alpha isotope after tumors become resistant to the beta isotope. And that's shown in the swimmer plot on the left and the waterfall plot on the right. This is from a group out of Germany. It's the very same construct and this very same molecule. The only difference is literally one atom. It's amazing to me that the difference between a patient potentially losing hope because of the treatment resistance or becoming responsive by changing the isotope from lutetium-177 to actinium-225. Now, that's not to say that there aren't challenges. When we want to design truly new de novo radiopharmaceuticals using constrained peptides, there are potential issues which we'll have to overcome. I've listed a few of them here for discussion, but in the interest of time, I'll just note that renal reuptake and retention can be a potential issue for peptidic or protein-based targeting moieties, because the kidney may readily metabolize these agents, and there are many reuptake mechanisms in the kidney. You would not believe how many there are. So it's a multifactorial issue. So hopefully I've convinced you that the potency of alpha particles and the potential utility of using diverse peptide libraries to find de novo chemical matter is useful. The key element of a successful drug development campaign in this field, I believe, is biodistribution, and then biodistribution. So thank you so much for your kind attention. And I'd also like to thank my colleagues and the scientific team at RayzeBio, as well as our funders and collaborators, and a special shout out to my colleagues, Aron Kantoff, Aron Knickerbocker, and Thomas Harding. Thank you so much.